Are you going to do the timing?
Yes.
Okay.
When she starts talking.
Okay.
Actually, doing it two timing. I'm doing funny stuff.
Okay.
Got it. Okay, relax.
Okay. Guys. It's a rowdy bunch. Okay, everybody. Hey, welcome to the December edition of the Lightning Talks. We have a pretty sweet lineup today, starting with Christine Choirat, who is everybody's favorite reproducibility expert. And she's promised me that at the end of her talk today, we're all going to be fluent users of GitHub. So.
And bilingual in French.
Yes. Welcome, Christine.
Thanks. So, I'm a data scientist. I do data science in environmental health. So, you've seen these diagrams. You're a hacker, you're a mathematician, you have substantive expertise, you're the unicorn right here. So that's the usual take. My take is more simple, so let's go very blunt. So there's data, there's science, and that's what I do. So if you want more literature about data science, you can actually read the real stuff. So it's pretty much... I've been working with Francesca and Corey Ziegler used to be here, and a bunch of people in this room on air pollution, right? And so that's the Eiffel Tower, so that's the French, this is what we see in Paris actually very often. And after 6 years working on air pollution, my take-home message is that it's bad for you. It's bad. It's bad. Alcohol is more ambiguous.
Cheers.
Within my term soon. So what I've been studying with a lot of data is how, energy is produced. The fact that you produce energy, you might burn coal. It generates sulfates that turn into some stuff that Lucas can describe, like particulate matter that contaminates the air. Then you breathe the thing, and then it's not good for you. So that's the long story. And we have a lot of data to study that, and we have a lot of data, which is why data science is needed in this case. We have Medicare, so we're talking about a cohort of like 500 million people a year, like it's very big. We have air pollution monitors, we have power plants, and we use complicated software. People in this room use complicated software to track what emits what breathes, etc., etc. But what I wanted to talk to you about in really 2 minutes is that one of the big points of data science is to make sure that the science that we generate is reproducible. So that when we publish a paper, we do have the code that corresponds to the paper, we do have the data, we can link the code, the data, the articles, and that we comply with all the security requirements if we work with health data, if we work with cohorts or claims data. So what we try to do is we try to force everybody to use tools like GitHub that I highly recommend to you. We make sure that the health outcomes are stored in very secure locations, and we make sure that we can reconstruct our own articles, and then we can share all the workflow with other people, right? So that's my job. That's what I do with people, so this is me. And, this is me, these are the postdocs, the PI here. Yeah, so it's cat herding. So because everybody is extremely productive, very smart, etc., but it's very hard to put together workflows that are going to be stable for 10 years. So that's my job. Right? And actually, so I still have 2 minutes. Just to tell you that after like 6 years here, I'm going to Switzerland to try to do the same thing, not for a department, but for a country. So the joke is that when it's not compulsory, it's prohibited. So that's what French people say about Switzerland. I hope there are no Swiss here. And instead of and there are big principles. And we provide, I will provide soon, and I will be working with people here, a platform to try to make reproducible data science at scale for like a whole country. So it's called Ranku, and you can give it a try. And this is the amazing team that I had the pleasure of working with for several years. And thanks, and I'm done. 4 minutes.
We just had
Where was Ranku developed?
It's developed, so it's in Switzerland. it's developed, so the academic director is the founder of VMware, and the team was hired directly from IBM Watson. And so it's developed in Switzerland by a team of about 20, 30 people. It's open source, it's deployable as a federation of deployments, so you can have one deployment in New York, one in Boston, and they talk to each other, sharing metadata. Everything is under version control automatically. Users don't have to learn Git. We track you. So, yeah.
So, Ranku is Japanese for Big Brother?
Yes, but it looks much nicer with the nice, when it's written. You're like, yeah, that's the thing. But you decide who has to be involved and what you want to share. So the idea is to provide a platform where people can opt in. We don't force you. It would be the other direction. In what we're doing now is like we say, we want your code on GitHub, we want your data in a repository, and this is an active effort of every researcher, every PI. The idea would be, as a federation of university environments or industry environments, we provide a platform, you're free to use it. If you use the platform, then data, code, workflows are under version control. That's the idea.
How much code and data can we put on there to do it free?
So it's a deployment thing. So you can totally have a deployment. There's a deployment at the Swiss level. In the Swiss cloud, we have unlimited budget resources, as far as I understand. So this is what they say until they say, not possible anymore. This is and it's really where you put it. If you put it on a gigantic Amazon instance, you have whatever you pay for. The software itself has no limitations. On data storage, you can connect like Amazon buckets, you can connect like any type of, it's a very, very thin middleware actually. So yeah. Yeah. Yes, 6 years. And you know that Marcello speaks French better than I do. Yeah, 6 years actually, 6 years here.
You speak English better.
Okay. Any more questions? All right. Thank you, Christine. From experience, I highly recommend working with Christine and having her double check everything you do, because she's the best at it. So our next talk is going to be by Kimberly Glass, who is a professor at the Brigham.
Stand over here. Yeah, just to be like, block all my slides. Yeah. Sounds good. And you can use this. Excellent. And forward back. Excellent. Right. Someone timing me? Oh, yeah. Excellent. All right. So I'm Kimberly Glass. I'm an assistant professor at the Channing Division of Network Medicine over at Harvard Medical School. Before I get into the science of my talk, I will learn how to use technology first. Does this count against my time? No. There you go. So, first off, a little bit about me, because I think a lot of people in the room probably don't know too much about me. I grew up deep in the South in Austin, Texas. And when I was young, I was really ambitious. So when I went to college, I decided I would get a Bachelor of Arts in Liberal Arts because I wanted to learn about everything. But I knew that wasn't practical, so I also got a degree in physics. For my graduate school, I kind of wised up. What? Practical physics? Come on, guys. That's better than liberal arts. For graduate school, I was wiser. I'll put that in quotes, top audience. And I decided to go and get a PhD in physics. After getting my PhD in physics, I came up here to Boston, where I'm still at today. I actually did my postdoc together with John Quackenbush and GC1, who you guys probably know at Dana-Farber and here at the School of Public Health. And now, as I said, I'm assistant professor at Harvard Medical School, which leads to two questions. Number one, why am I getting farther and farther away from nice, warm weather and tacos? And secondly, what is a physicist like me doing at a medical school? Well, there's two main reasons. One, good advice and training, and these are two of my graduate advisors. I could talk a lot about how they steered me in the right path, but since I know we have a lot of grad students here, listen to your mentors, listen to your advisors. They give you good advice. They steered me towards this field. And secondly, the reason I'm here is because what I call the biomedical data revolution. And you can really tell this by looking at when I completed my PhD. Around 2001 is when the Human Genome Project was completed. You can see just data increased. And so I got really interested in how to analyze this data. Now, as a physicist, I walked in the room, and this was my conception of what DNA looked like. It's a string. Everything we need to know is on this string. And as I learned more and more and more, I realized it wasn't quite that simple. Along this string are various sequence elements that do important things like recruit transcription factors. Transcription factors don't act in isolation. They can form these complexes. There's changes along the DNA, which are both genetic and epigenetic. Those are variants and CG methylation. Of course, there's also the 3D structure of the DNA. You have the chromatin state. You have how things cross-link, things like that. The end result of a gene isn't a gene, right? It's actually mRNA. Now, mRNA isn't the end of the deal. The mRNA can actually post-translationally modify by a microRNA and ultimately creates a protein. Now, the physicist in me looked at this and said, this is really complex, but I believe in spherical cows. Therefore, let's draw this instead, right? And this kind of defines my research. I've been spending the last couple of years looking at biological networks. And so why are biological networks important? Why is what I showed you important? Well, biological networks are important because biological processes, how one gene talks to the next, can be encoded in these biological networks. One way to think about this is through a special type of network that I focus on called a gene regulatory network, in which the nodes are the genes, and the edges between them are causal relationships between those genes. And finally, a lot of the things I do is about comparing these biological networks. The fact that someone with a disease and without a disease have different networks. The fact that different men and women in this room have different networks. And how do we compare these things? So how do we actually do that? Well, we have a network, and out of the network comes a lot of different types of omics data. And of course, this omics data is really complicated. I showed it to you. And I like to think about it as a lot like the blind man and the elephant. Everyone sees a different aspect of the elephant. Different types of omics data represent different types of underlying biological network. So what we ultimately want to do is we want to take this omics data and reverse engineer and try to estimate what the network was that could have produced this data. Now, several years ago, I came up with an algorithm that's kind of defined my research over the last 5 years to try to do that. And this algorithm is called Panda. It stands for Passing Attributes Between Networks for Data Assimilation. But more importantly, it's a cute, fuzzy animal that everyone can remember and pronounce. So from now on, I'll call it Panda. And it takes in 3 types of omics data. It takes in protein-protein interaction data, protein-DNA interaction data, and gene expression data. It has these 3 data types as 3 types of networks. Again, a PPI network, interaction between transcription factors and genes, and gene co-expression or gene co-regulation, basically how connected 2 genes are in their expression levels. It layers these onto each other, making 3 hierarchical structure of the networks, and uses message passing to find consistency across these. Now, there's not nearly enough time in 5 minutes to go through the mathematics, but at a high level, we basically take information from these outside networks, pass to the inside, and back and forth. Is that my 2-minute warning, or is that just...
That's your 5-minute warning.
That's my 5-minute warning, and that 2 minutes left.
You're done.
Great. I can wrap up in 2 minutes. All right. So to wrap up really quickly in my next 2 slides, because I have 2 slides left, I showed this really complicated picture at the beginning. And you notice if you looked at Panda, there were only 3 types of omics data in there, and I talked about how important that is. What about all the other types of omics data? Now, obviously, we can't just stick to 3. There's a lot more in there. But I'm beginning to develop what I call the network zoo. And I can't go through all these algorithms today, but at a very high level, I've talked to you about Panda, which is how to reconstruct gene regulatory networks. I've been working with my postdoc Abhijit on Spider to model epigenetic networks, Marika Kujer to model microRNA networks, Linus to build individual sample networks, Condor to characterize the structure of networks, Alpaca to compare structures, and Monster to identify network rewiring. And you'll probably see a theme here. Cute, fuzzy animals that everyone can remember the name of. So, briefly to conclude, biology is complicated, data is really complex, but we can use networks as a powerful way to try to model the processes in the cell. And finally, if we want to understand the systems in biology, we need to develop methods, not just one method, but a host of methods that can actually do these things. Thank you. Did I talk quickly?
How long do you spend on coming up with the names?
It's an active debate every time there's a new method. And sometimes they change names over time. Yeah. It's actually a lot of fun, though.
What are some failed names?
Red panda. Spider used to be called red panda, and then we decided it was too similar to panda. We have an algorithm that we're thinking about naming Cayman, which we'll see if that sticks around, and one called Raven, we'll see if that sticks around. So, haven't had too many failed names, but we definitely have some like temporary names as placeholders.
The acronyms are very hard for W.
I'm sorry to keep riffing on the acronym thing. Do you have a Python version of Panda, and is any of it written in Pandas?
There is a Python version of Panda. I didn't implement it. I think it might leverage Pandas, but I don't know. But there's a Python version. You can download it and check for me.
Awesome.
See if it's loopy.
Okay.
Can I stand at the screen?
Oh, yeah, that's fine.
All right.
Oh, sorry, I'll open that. We will see you.
All right, our next, thank you again, Kimberly.
I'd like
Our
to
next
look at the
speaker
screen one
is going to be Alkes Price, talking about genetic architectures of human diseases and complex traits.
My next stop, Montreal.
All right, good afternoon, everybody. I'm Alkes Price. The title of my lightning talk is Genetic Architectures of Human Diseases and Complex Traits. I'd like to start by answering a few questions that I'm sure you're all wondering about the answers to, starting with who is Alkes Price? Well, to state the obvious, we are what we publish, so I've parsed out some of the titles here of papers trainees in our group have published this year. You'll see things about disease heritability, mixed model association disease architectures, and particularly functional architectures and functional enrichment, which is increasingly a theme in our group as we seek to understand functional components of disease and complex trait heritability. Now, the next question I'd like to answer is, what does genetic architecture mean? So we might be talking about different flavors of heritability, SNP heritability or narrow sense heritability. How many causal variants are there impacting disease? How are those causal variants distributed across coding or non-coding regions, or common versus rare variants, or particularly across different functional annotations? And the last question I'd like to answer is, why should we care about genetic architectures? Well, understanding genetic architectures of diseases and complex traits can help us understand disease biology and biological aspects of evolution. Or more pragmatically, it can help us increase GWAS association power or increase polygenic prediction accuracy and improve fine mapping resolution. So to give some examples, I have 3 short stories I'd like to tell during my 5 minutes. The first is a story about tissue-specific trait biology. So way back in 2015, we developed a new method called stratified LD score regression and applied this method to histone marks in specific tissues and cell types to obtain statistically significant evidence. Now, these findings are not surprising, that for schizophrenia, it's brain tissue that is somehow driving the disease. For height, it's connective bone tissue that's most important for that trait. And for rheumatoid arthritis, it's immune cells. Well, in more recent work published this year, we've undertaken a completely different approach by looking at specifically expressed genes and signals of significant excess heritability lying in or near specifically expressed genes that are specifically expressed in particular tissues or cell types. And we got the same signals for schizophrenia, rheumatoid arthritis, and height, but for some other diseases and complex traits, in some instances, the evidence that we got was complementary, suggesting that both chromatin data and specifically expressed genes can be informative for tissues and cell types. The next story is a story about functional architecture of low-frequency variants. So as we move to whole genome sequencing data, people are increasingly interested in what's going on with rare and low-frequency variants. So on this slide, I have a plot of both low-frequency variant functional enrichments and common variant functional enrichments across various diseases and complex traits. Now there's about 55 light blue dots on this slide that all represent brain annotation, brain trait pairs in which the low frequency variant functional enrichment is more than twice as large as the common variant functional enrichment. One of these points is circled, and this circle point is telling us that H3K4ME3 marks in brain prefrontal cortex explain 57% of the low frequency variant heritability of the brain-related trait neuroticism versus only 12% of the common variant heritability. Now, why is it that we have these instances where certain functional annotations are explaining a lot more low-frequency variant heritability proportionately as compared to common variant heritability? It turns out that the answer has something to do with negative selection. And using forward simulations that I will not describe in detail, we determined that this ratio, the LFVE versus CVE ratio, is somehow an indicator for the strength of negative selection. And what this means is that variants that impact the risk of brain-related diseases or traits are somehow more impacted by negative selection. Now, this might be pleiotropy selection. You might have a variant that either increases or decreases the risk of schizophrenia. We know that schizophrenia decreases your fertility, but you might either increase or decrease your risk of schizophrenia and affect the brain in some profound way, and that variant might thus be deleterious. And that's why we're seeing these signals of brain annotation trait pairs. The last story I'd like to tell is a story about polygenicity. So every geneticist knows that common diseases and complex trait are extraordinarily polygenic. Where you run a GWAS, you get a Manhattan plot with zillions of common trait associations, usually explaining not very much heritability. Why are common disease and complex traits so polygenic? Well, in unpublished work, we have a hypothesis that we call the flattening hypothesis, in which this common variant polygenicity arises because negative selection flattens common variant effect sizes. So on this toy diagram, let's start with the gray shaded areas. The gray shaded areas are de novo mutations. De novo mutations, you'll see some very large effects at important genes or at important regions, such as coding regions within those genes. But if you look at common variant effects here in dark orange, common variant effects are always small. And that's because if a variant does something, it's probably bad for evolutionary fitness, and a negative selection in evolution is just not going to allow it to increase to high frequency to being common variant. So if you see, if a variant increases to being common, it's probably a very small effect, and you get a flattening across the board where it doesn't matter if you're an important gene, unimportant gene, coding region, non-coding region, the variant effect sizes are always very small, and that reduces the polygenicity. Indeed, in empirical analyses of a large set of diseases and complex traits, we consistently saw that common variant polygenicity is about 4 times more than low frequency variant polygenicity. All right, I'm finishing. And our evolutionary modeling tells us that it's about 30 times more than de novo variant polygenicity. Interestingly, the most polygenic trait here is number of children, indicating that, not surprisingly, the number of children that a person has, may be related to the processes of evolution. I'm done with the scientific portion of this talk. I do want to take my last few seconds to advertise the course that I'm teaching in spring term. This is the entire spring term, EPI 511, and this is a course that is very commonly taken by many biostatistics students. This year's TA is a biostatistics doctoral student, Margot Hujuel, and please feel free to ask me any questions you may have about this course. Thank you.
I'm curious, when you talk about the flattening of the effect size, is that because negative selection enriches for small effect sizes?
So the key question is, what does it take for a variant to become common? If a variant is deleterious, evolution will not allow it to become common, because evolution will keep pushing down the allele frequency so that it cannot become common. So if it becomes common, that means it must have a tiny effect. And so you get tiny effects. It might be a tiny effect in an unimportant region of an unimportant gene, or it might be a tiny effect in a really important region of a really important gene. But either way, the effects are going to be tiny across the board. And that's the sort of flattening that you don't see if you look at de novo variants. Or for de novo variants, because they haven't been subject to the pressures of negative selection, you might see some really large effect sizes.
And do you see a correlation between the processes affected by small effect size variants in particular traits and the rare variants with big effect sizes, right? So are they hitting the same pathways? Are they affecting the same genes?
We believe that in general, we haven't tried to answer that question. We suspect that they're hitting the same pathways and the same genes. But the point is that if you see a common variant, you know that it doesn't have a large effect. It could be either having a small effect on a gene that turns out to be really important, or it could have a large effect on a gene that turns out to be not so important. And we think it's more interesting to search for variants that have small effects on genes that turn out to be really important. And one of the implications of this is that if you run a GWAS and you get a locus and then you find map it and you find a coding variant, people tend to think that's pretty exciting, but we think that's pretty unexciting. Because if you have a large effect on a gene and it's a common variant, then that means it's probably a really unimportant gene in order such that the aggregate effect size is small. Yeah.
Question. So when you look at the location of those variants, do they get around the common variants? Just because I can imagine that at the protein levels, when you have actually one variance that's like small effect, that if you have a bunch of variants that have big effects, that will actually follow through small variants that actually compensate.
Yeah, your question is whether rare variants rare or low frequency variants that contribute to heritability tend to localize close to common variants that contribute to heritability. I don't know the answer. I think it's a very interesting question. There's a related question that is sort of on our radar screen, which is that do common variants that contribute to heritability, do they concentrate or cluster in specific regions of the genome? And we certainly know that with respect to certain functional annotations, the answer is definitely yes. And with respect to certain gene sets that are enriched for heritability, the answer is definitely yes, although we're trying to answer that question in more broad ways. And if we figure out how to do that, we may also be able to answer the question that you asked, although we haven't answered it yet.
Some interesting questions you could ask about comorbidities in these low frequency variants.
Yeah.
Okay. And our next speaker is going to be a postdoc here at
Office mate.
Yeah, one of my office mates, Luwan, who works with Tianxi, right?
Yep.
Thanks, Luwan.
How to work this?
Turn it on.
Is it working?
Turn it on.
Oh,
There's an on click on the side.
Yeah, on this thing.
I had to go.
There you go.
don't count the time
Yeah,
yet.
sure.
Yeah.
Yeah. Okay. Yeah.
Okay. Before I start, I'm really impressed by Alkes' speed. So impressive. So impressive. I wish I could do that, but I couldn't. So it's possible for me to overtime. Yeah. All right. So my name is Luwan, and I'm working with Tianxi on electronic health records. So today I'm going to talk about some recent developments on unsupervised and ensemble learning methods in EHR-based research. So as we all know that electronic health records really provide tremendous opportunities for research because it's really stored a lot of rich phenotype data, including not only codified information, for example, diagnosis, medications, procedures, and lab results, but also more importantly have a lot of meaningful clinical concepts extracted using natural language processing from tons of narrative texts. And more than that, we really have the luxury to get access to large sample size. However, with these advantages I have mentioned above, we actually have some pressing challenges as well that really needs to be addressed before truly enabling EHR for research. And today I'm going to talk about two most important challenges. The first challenge I want to touch base on is about heterogeneity, which can be often observed by the fact that a lot of medical concepts and medical codes will have been used differently across different EHR systems. And patient populations differ a lot from one hospital to another. So such a heterogeneity really will lead to power loss and will contribute as a huge barrier when we talk about reproducibility and information synthesis. So that's the first challenge. The second challenge is about noise. So when you talk about a given phenotype, and then you want to build a model to predict the risk for that phenotype, and then chances are you can collect tons of features you want. But more often than not, most of them turns out to be very irrelevant or very redundant. And then the question here is, how can you get rid of those junk features that will ultimately lead to a poor performance? So that's the key. So I would dive a little bit detail into these two aspects to just give you a little bit of flavor what we have done. So for heterogeneity, like I mentioned, a lot of medical concepts or codes have been used differently. The reason behind this really being that a lot of codes have many synonyms. And turns out they actually have some successful, well-established manual grouping procedures, for example, the FICOs or UML's CUIs. But those kind of manual approaches are really not scalable and subject to human bias. So our question here is, how can we develop a data-driven and automated approach that really can be scalable and get rid of the human bias? And our solution here is the multi-view spectral clustering. Multi-view really encode the multiple data sources that are really prone to heterogeneity, and spectral clustering is really one of the most commonly used techniques in network analysis in machine learning community. This plot actually shows you how different across these three data sources we have. You can see the huge, humongous differences in these data sources. So that really signify the need to find a weighted average instead of like a simple average. So that's pretty much what I want to say about heterogeneity. So next I'm going to talk about a little bit about noise. So for noise, for a given phenotype, if you want to predict the risk of a phenotype, you can collect many features from your EHR. For example, your ICD count or some your CUIS counts or even healthcare utilizations. So among all these features, how can you identify the experts? That's the key question. Because if you include many of the junk features, you end up with a very poor model. Then that's useless. So this one actually shows you this one actually is the plot in terms of AUC about each individual feature. And then you can see most of them really behave very bad. And sporadically, you can see a few of them can above AUC above like 0.8. So that really indicates the need to tease out the experts. So the question for us is really to how can we discern and aggregate experts, and so that we can boost the predictive performance in the end. The solution here is we leverage a probabilistic model, so-called icing model, and we try to recover the edges so that we're able to locate to a very local structure that only involves the experts. By doing that, we can build our model to boost the performance. This is my last one. I finished. So this is our model, and then you can see this is our I don't know how to point it. So that's the top one. Anyway, the top one is our result. And then we're able to identify the experts colored in red. Yeah, I finished. Thank you.
Questions for Luwan?
So you talked about heterogeneity between EHR systems and between hospitals. What about between doctors within the same hospital?
Exactly. The heterogeneity, the first part of the different coding behaviors also include like the different physicians' behaviors as well. Yeah.
And also, do you ever run into any issue with the fact that the codes are made for billing purposes rather than actual diagnoses?
Exactly. I intentionally want to put a figure that how different like the diagnosis codes for billing purpose. Like say they all meant to be the same phenotype, but just slightly difference in your body parts. Say your right left race or right race, something like that. But I feel like because of the time limit, I just gave up that idea. But we do actually group those kind of codes together as a whole group, so that we can actually enhance the signal. Yeah.
But for acute injuries, that's probably okay, but for chronic conditions, it might not be, right?
Mm-hmm.
And I think that one of the things I worry about is that any individual encounter, so anytime you go to your physician, something may get coded for something, but it may have actually nothing to do with your health status. Right?
Yeah, exactly. So that's why when you only consider about your healthcare utilization, it's not like it's kind of partial. So that's why this feature is useless. So when we do that, we want to really get rid of those. But before that, you don't have the information.
Yeah, I mean, I always tell the story. I talk about EHRs, where when my son was young, he was about vertigo, and I went to the emergency room. They didn't know what was happening. They did EKGs and EEGs. And they said, oh, it's a benign condition. Go home and be fine the next morning. And I was fine. And the next morning, my physician called me to follow up on the heart attack I had the night before, because they did an EKG, so they said, oh, suspected heart attack. Right?
Yeah.
So you get these weird things, and so do you follow these, and if cardiovascular events turn up in 4 or 5 subsequent visits, then you might say I have a heart condition. But if it shows up once, it may just be that noise.
Yeah, that's true. Yeah, so
How do you handle that kind of situation? Do you look for correlations across encounters?
what we did is actually in this graph, basically you can see this graph allows your dependence. So for example, you can see a single node as just using your encounter number, and then the other node can be, you can do some normalization so that you can get rid of like the just by visit many times, but just turn out to be useless. But conditioning on your encounter number, they may be independent, but if you do include the encounter number, they are dependent. So that's why we want to build this kind of dependent structure so that you can still borrow some strength. Yeah. Because the more visit, you may more likely have the kind of thing. Yeah.
My heart's
Sorry about that. Yeah.
Any more questions? Okay, let's thank Luwan again.